Hello, everyone. My name is Fatima Romero Castillo. I am a master's student in chemistry at the Metropolitan Autonomous University Unit Iztapalapa in Mexico City. My research project is genetic engineering of phage M13 to design diverse phases of liquid crystals. As an introduction, recently there has been more interest in using biomolecules like DNA, proteins, and viruses in the creation of new materials. This is because they have a well-defined structure and useful chemical properties. These biomolecules and their features are suitable for designing materials used in technology. Specifically, the M13 bacteriophage virus is harmless to humans and only infects bacteria. It has a pencil shape with a diameter of 6.5 nm and a length of 880 nm. Its 5 most important proteins are P3, Fur, 7, P9, and PATE. The PATE protein makes up 85% of the virus's mass because it covers the virus body, protecting the single-stranded DNA made of 6,407 nucleotides. This makes the PATE protein the main part responsible for the bacteriophage's properties. One quality of the bacteriophage is its ability to self-assemble into liquid crystals with different phases, like the nematic phase and the cholesteric phase, as seen in Figure 2. This depends on concentration, pH, and other experimental factors that affect how it assembles. Since the PATE protein is the main participant in the self-assembly of the bacteriophage through interactions between viruses, it is crucial for forming liquid crystals with different phases. In this work, we genetically modified the PATE protein sequence by changing the tyrosine at position 21 to methionine and tryptophan. The methods used to create the mutants were directed mutagenesis of the PATE protein using the M13 plasmid. We used specially designed primers that have the desired mutant sequence. We synthesized using DNA polymerase, then we did digestion and transformation to get plates with the mutant DNA. After getting the plates, we grew and purified the phages with mutations in PATE. They were allowed to grow at specific temperature and time, followed by centrifugation and precipitation with Phage and NaCl. With the obtained phage, we used 2 methods to get liquid crystals. In the first method, we added a certain amount of phage into a capillary, controlling concentration and temperature, and let them sit for several days. We also used the dip coating method, which involves dipping a glass substrate into a solution of bacteriophage under controlled conditions like pH, concentration, and speed. The DNA obtained after purification was sent for sequencing to make sure we had the desired mutants. When we saw that the change from tyrosine to methionine was successful, we proceeded to do further analysis. We conducted a circular dichroism study to ensure that the secondary structure of the PATE protein, which is an alpha helix, did not change with the introduction of the mutant. From this analysis, we confirmed that there were no changes in the secondary structure when compared to the native phage, ensuring that this structure remains stable. Results of M13 phage self-assembly and film formation. This study focuses on understanding the liquid crystal phases of the M13 bacteriophage and its mutants using optical polarization microscopy, POM. We look at how temperature and storage time affect the self-assembly of the viruses. The results show that temperature and storage time have an impact on the formation of liquid crystal phases in the M13 bacteriophage and its mutants. The differences seen between the 1M I21W1W mutants suggest that changes in the virus structure can affect how it self-assembles. For the dip coating method, we present images taken with optical polarization microscopy, POM, of films made by the M13 bacteriophage under different solution conditions and extraction speeds. The results indicate that the extraction speed significantly affects the organization of the mesomorphic phases of the M13 bacteriophage, showing different self-assembly patterns that could be important for biotechnological applications. In conclusion, our observations suggest that the I21W mutation does not significantly change the protein structure while the I21M mutation affects the self-assembly process. This is important for understanding how these bacteriophages behave and their potential uses in biotechnology and nanomaterial.